Fenestrated.
Yeah, because it's fenestrated, which means it is porous. So little holes between the epithelial cells of the capillaries. And then in addition, we have those podocytes forming the visceral membrane covering it, and they have those little filtration slits between their petted cells that allow materials to filter out, right? So that's our filtration membrane. All right, so there will be some questions on the exam. Does anybody have their study guide for the lab exam?
I do.
Because there are some questions, if I remember correctly, I didn't look at it just before I left today, but there are some questions that have to do with how the kidneys help to regulate blood pressure and stuff. So,
I read it somewhere.
all right. So those were the.
Yeah. Okay.
So we already talked about the parts of the nephron. So we have the renal corpuscle, the renal tubule, the proximal convoluted tubule, lubricantly, distal convoluted tubule. We already talked about what happens in each section. And what happens during glomerular filtration. We already talked about what's being filtered out, what's not being filtered out. What is a normal glomerular filtration rate value? 125 milliliters per minute, right? That's normal for males, but we're just going to go with that value. It's a little less for females. What physiological factors maintain glomerular filtration rate homeostasis despite changes in body conditions? And we just talked about that, how the afferent arteriole can be vasoconstricted, can be vasodilated under different conditions, how the juxtaglomerular cells can produce renin if blood pressure starts to drop to raise the blood pressure and things like that. And we already talked about what region does the majority of reabsorption take place. What was that? Proximal convoluted tubule. And we talked about what components of plasma filtrate are normally reabsorbed. Well, yeah, we didn't really go into, there's other stuff here about, but the stuff that I think you already know what the normal constituents of urine are. You don't need to know the actual percentages or anything like that. But what would be considered a normal substance in the urine? What would be considered abnormal, like, glucose, ketones, things like that would be abnormal. And what disorders are indicated by the abnormal components. We talked a little bit about that in class. Also, in the chemical and physical processes of digestion, you should know what amylase is, what it digests, what's the optimal temperature and pH for amylase to function. pH is normal body pH, around pH of 7. Optimal temperature is body temperature, 37 degrees Celsius or 98.6 degrees Fahrenheit. What two organs produce amylase?
Salivary amylase.
Salivary gland, salivary amylase, pancreatic amylase from the pancreas, right? What is pepsin?
Stomach.
It's an enzyme that's produced by the stomach.
To digest protein.
And it initiates the digestion of protein in the stomach. What's the optimal pH for pepsin to function?
It varies.
What's the pH in the stomach? Usually about a pH of about 2 or so, between 1 and 2. So that would be optimal pH. Could you take some trypsin, which is a protein digesting enzyme in the small intestine, and if the person wasn't producing pepsin, could you put that in the stomach and have it work?
No.
No, because in the small intestine we have alkaline bicarbonate that's secreted there to neutralize the acid. So its optimal pH is going to be closer to the pH of the small intestine, which is close to 8 or so. What is lipase and what does it digest? Fats. And what's the optimal pH for that lipase to function?
7.
Yeah, right around 7, between 7 and 8. How was lipase digestion determined in our experiment that we did? By the production of
Oily.
fatty acids, and we measured the pH, right? Remember we had that litmus cream, and when we had the generation of fatty acids, the cream turned kind of a pink color as opposed to its normal kind of plain color, right? What's the main organ that produces lipase? Pancreas. There is salivary lipase, but it doesn't really do very much. Is bile an enzyme?
No.
No. What organ produces it?
Liver.
The liver. And it's stored in the gallbladder. Now, what is bile's digestive function?
Emulsification.
To emulsify fats, and what does it mean to emulsify fats?
Break up to
Break it up
milk.
into tiny little
Tiny.
droplets so we have greater surface area for the lipase to work on, right? Now, we did that experimental design where you designed your own experiment to determine whether we had carbohydrates, fats, proteins, things like that. So what I would expect you to know from that is what chemicals do we use to test for the presence of carbohydrates? What chemical do we use for the test for the presence of proteins? What chemical do we use, or we didn't use a chemical actually, we did the lipid spot test to test for lipids. And also, what constitutes a positive and a negative result? So what is the chemical we use to test for the presence of monosaccharides or reducing sugars? Benedict solution. What do we have to do with the Benedict solution?
You have to boil it.
We also have to boil it, right? And what chemical did we use to test for the presence of starch? IKI, iodine. And what was considered to be a positive test for Benedict? For sugar?
Green or
orange to green, somewhere around in there. Rusty, orange, green, etc. How about what's considered a negative test for sugar when you put Benedicts in it?
like blue.
It stays the same blue color of the Benedicts.
Oh, but it was tan.
No, tan is with the iodine. It stays tan if there's no polysaccharides. And what color does it change if there are polysaccharides or starches?
Blue-black.
Blue-black,
Blue
so
black.
dark colored, right? For proteins, what were the chemicals we used to test for the presence of proteins?
There were different ones.
Biuret solution and sodium hydroxide. We used
That was
BAPNA
for
just
the
in the
lab in class.
the virtual lab. But in the real wet lab, we used biuret solution and sodium hydroxide. And what color did it turn if there were proteins in it?
Purple?
Purple. And what color did it stay if there's no proteins? Just the light blue of the biuret solution. How do we test for the presence of lipids? Spot test.
Spot,
We
yeah.
just put some little spots on the thing, and what did we see if there were lipids in it?
Oily
Stayed as
residue.
oily stain, and if there weren't any lipids, it just dried out, right? Be sure you know how to design an experiment to test for the presence of those different things. I might have a question on the exam that asks you, we want to know if this is present or something. How would you design an experiment to test for that? Be sure to be able to include positive controls and negative controls. What's always a negative control? Water, right? We know water doesn't contain any of those things. Doesn't contain protein, doesn't contain lipids, doesn't contain starches, whatever. And then what's going to be a positive control?
The item you're testing.
Something that does contain what we're looking for, right? That's going to be your positive control. So be sure you know what we mean by positive and negative controls. And then our lab that we did today, acid-base balance, we just kind of went over all that stuff. The difference between respiratory acidosis and alkalosis versus metabolic acidosis and alkalosis. The causes of respiratory acidosis and alkalosis, and how the renal system compensates for those conditions, and then the causes of metabolic acidosis and alkalosis, and how the respiratory system compensates for those things. Be sure you know that equation. Carbon dioxide plus water yields carbonic acid, which splits apart into bicarbonate and hydrogen ions. Now know under what conditions the reaction would be shifted to the right, and under what conditions the reaction would be shifted to the left. Okay? All right, I think that's about it. And what the normal pH range is in human blood is about 4.5